Chapter 32 Plant Reproduction 32.1 Reproductive Development and Structure. By the end of this section, you will be able to do the following. Describe the two stages of a plant's life cycle. Compare and contrast male and female gametophytes and explain how they form in angiosperms. Describe the reproductive structures of a plant. Describe the components of a complete flower. Describe the development of microsporangium and megasporangium in gymnosperm. Sexual reproduction takes place with slight variations in different groups of plants. Plants have two distinct stages in their life cycle: the gametophyte stage and the sporophyte stage. The haploid gametophyte produces the male and female gametes by mitosis in distinct multicellular structures. Fusion of the male and female's gametes forms the diploid zygote, which develops into the sporophyte. After reaching maturity, the diploid sporophyte produces spores by meiosis, which in turn divide by mitosis to produce the haploid gametophyte. The new gametophyte produces gametes, and the cycle continues. This is the alternation of generations, and is typical of plant reproduction. Figure 32.2. Figure 32.1 Plants that reproduce sexually often achieve fertilization with the help of pollinators such as A. Bees, B. Birds, and C. Butterflies. Credit A. Modification of work by John Severns. Credit B. Modification of work by Charles J. Sharp. Credit C. Modification of work by Galloweb. Design, Flickr. Introduction. Plants have evolved different reproductive strategies for the continuation of their species. Some plants reproduce sexually and others asexually, in contrast to animal species, which rely almost exclusively on sexual reproduction. Plant sexual reproduction usually depends on pollinating agents, while asexual reproduction is independent of these agents. Flowers are often the showiest or most strongly scented part of plants. With their bright colors, fragrances, and interesting shapes and sizes, flowers attract insects, birds, and animals to serve their pollination needs. Other plants pollinate via wind or water, still others self-pollinate. Chapter Outline 32.1 Reproductive Development and Structure 32.2 Pollination and Fertilization 32.3 Asexual Reproduction. Figure 32.2 The alternation of generations in angiosperms is depicted in this diagram. Credit. Modification of work by Peter Coxhead. The life cycle of higher plants is dominated by the sporophyte stage, with the gametophyte born on the sporophyte. In ferns, the gametophyte is free-living and very distinct in structure from the diploid sporophyte. In bryophytes, such as mosses, the haploid gametophyte is more developed than the sporophyte. During the vegetative phase of growth, plants increase in size and produce a shoot system and a root system. As they enter the reproductive phase, some of the branches start to bear flowers. Many flowers are born singly, whereas some are born in clusters. The flower is born on a stalk known as a receptacle. Flower shape, color, and size are unique to each species, and are often used by taxonomists to classify plants. Sexual reproduction in angiosperms. The life cycle of angiosperms follows the alternation of generations explained previously. The haploid gametophyte alternates with the diploid sporophyte during the sexual reproduction process of angiosperms. Flowers contain the plant's reproductive structures. Flower structure. A typical flower has four main parts, or whorls, known as the calyx, corolla, androecium, and gynoecium. Figure 32.3. The outermost whorl of the flower has green, leafy structures known as sepals. The sepals, collectively called the calyx, help to protect the unopened bud. The second whorl is comprised of petals, usually brightly colored, collectively called the corolla. The number of sepals and petals varies depending on whether the plant is a monocot or dicot. In monocots, petals usually number 3 or multiples of 3. In dicots, the number of petals is 4 or 5, or multiples of 4 and 5. Together, the calyx and corolla are known as the perianth. The third whorl contains the male reproductive structures and is known as the androecium. The androecium has stamens with anthers that contain the microsporangia. The innermost group of structures in the flower is the gynoecium, or the female reproductive components. The carpel is the individual unit of the gynoecium and has a stigma, style, and ovary. A flower may have one or multiple carpels. 890 Chapter 32, Plant Reproduction. Access for free at openstax.org. Visual connection. Figure 32.3 The four main parts of the flower are the calyx, corolla, androecium, and gynoecium. The androecium is the sum of all the male reproductive organs, and the gynoecium is the sum of the female reproductive organs. Credit. Modification of work by Mariana Ruiz Villarreal. If the anther is missing, what type of reproductive structure will the flower be unable to produce? What term is used to describe an incomplete flower lacking the androecium? What term describes an incomplete flower lacking a gynoecium? If all four whorls, the calyx, corolla, androecium, and gynoecium, are present, the flower is described as complete. If any of the four parts is missing, the flower is known as incomplete. Flowers that contain both an androecium and a gynoecium are called perfect, androgynous or hermaphrodites. There are two types of incomplete flowers. Staminate flowers contain only an androecium, and carpellate flowers have only a gynoecium. Figure 32.4. 32.1, Reproductive Development and Structure 891 Figure 32.4 The corn plant has both staminate, male, and carpellate, female, flowers. Staminate flowers, which are clustered in the tassel at the tip of the stem, produce pollen grains. Carpellate flowers are clustered in the immature ears. Each strand of silk is a stigma. The corn kernels are seeds that develop on the ear after fertilization. Also shown is the lower stem and root. If both male and female flowers are born on the same plant, the species is called monoecious, meaning, one home. Examples are corn and pea. Species with male and female flowers born on separate plants are termed dioecious, or two homes, examples of which are C. papaya and cannabis. The ovary, which may contain one or multiple ovules, may be placed above other flower parts, which is referred to as superior. Or, it may be placed below the other flower parts, referred to as inferior. Figure 32.5. 892 Chapter 32, Plant Reproduction Access for Free at OpenStax.org. Figure 32.5 The lily is a superior flower, which has the ovary above the other flower parts. B. Fuchsia is an inferior flower, which has the ovary beneath other flower parts. Credit a photo. Modification of work by Benjamin Zwitnig. Credit B photo. Modification of work by Koshi, Flicker, male gametophyte. The pollen grain. The male gametophyte develops and reaches maturity in an immature anther. In a plant's male reproductive organs, development of pollen takes place in a structure known as the microsporangium. Figure 32.6. The microsporangia, which are usually bilobed, are pollen sacs in which the microspores develop into pollen grains. These are found in the anther, which is at the end of the stamen, the long filament that supports the anther. 32.1, Reproductive Development and Structure 893 Figure 32.6 shown as a cross-section of an anther at two developmental stages. The immature anther, top, contains four microsporangia, or pollen sacs. Each microsporangium contains hundreds of microspore mother cells that will each give rise to four pollen grains. The tapetum supports the development and maturation of the pollen grains. Upon maturation of the pollen, bottom, the pollen sac walls split open and the pollen grains, male gametophytes, are released, as shown in the B micrograph of an immature lilianther. In these scanning electron micrographs, pollen sacs are ready to burst, releasing their grains. Credit A. Modification of work by Libre Texts. B. Modification of work by Robert R. Wise. Scale bar data from Matt Russell. Within the microsporangium, each of the microspore mother cells divides by meiosis to give rise to four microspores, each of which will ultimately form a pollen grain. Figure 32.7. An inner layer of cells, known as the tapetum, provides nutrition to the developing microspores and contributes key components to the pollen wall. Mature pollen grains contain two cells, a generative cell and a pollen tube cell. The generative cell is contained within the larger pollen tube cell. Upon germination, the tube cell forms the pollen tube through which the generative cell migrates to enter the ovary. During its transit inside the pollen tube, the generative cell divides to form two male gametes, sperm cells. Upon maturity, the microsporangia burst, releasing the pollen grains from the anther. 894 Chapter 32, Plant Reproduction Access for Free at OpenStax.org. Figure 32.7 Pollen develops from the microspore mother cells. The mature pollen grain is composed of two cells, the pollen tube cell and the generative cell, which is inside the tube cell. The pollen grain has two coverings, an inner layer, entine, and an outer layer, exine. The inset scanning electron micrograph shows Arabidopsis laurata pollen grains. Credit, Pollen Micrograph. Modification of work by Robert R. Wise. Scale bar data from Matt Russell. Each pollen grain has two coverings. The exine, thicker, outer layer, and the entine, figure 32.7. The exine contains spiropylenin, a complex waterproofing substance supplied by the tapetal cells. Spiropylenin allows the pollen to survive under unfavorable conditions and to be carried by wind, water, or biological agents without undergoing damage. Female gametophyte, the embryo sac. While the details may vary between species, the overall development of the female gametophyte has two distinct phases. First, in the process of megasporogenesis, a single cell in the diploid megasporangium, an area of tissue in the ovules, undergoes meiosis to produce 4 megaspores, only one of which survives. During the second phase, megagametogenesis, the surviving haploid megaspore undergoes mitosis to produce an 8-nucleate, 7-cell female gametophyte, also known as the megagametophite or embryo sac. 2 of the nuclei, the polar nuclei, move to the equator and fuse, forming a single, diploid central cell. This central cell later fuses with a sperm to form the triploid endosperm. 3 nuclei position themselves on the end of the embryo sac opposite the micropyle and develop into the antipodal cells, which later degenerate. The nucleus closest to the micropyle becomes the female gamete, or egg cell, and the 2 adjacent nuclei develop into synergid cells, figure 32.8. The synergids help guide the pollen tube for successful fertilization, after which they disintegrate. Once fertilization is complete, the resulting diploid zygote develops into the embryo, and the fertilized ovule forms the other tissues of the seed. 32.1. Reproductive development and structure. 895. A double-layered integument protects the megasporangium and, later, the embryo sac. The integument will develop into the seed coat after fertilization and protect the entire seed. The ovule wall will become part of the fruit. The integuments, while protecting the megasporangium, do not enclose it completely, but leave an opening called the micropyle. The micropyle allows the pollen tube to enter the female gametophyte for fertilization. Visual connection. Figure 32.8 is shown in this diagram of the embryo sac in angiosperms. The ovule is covered by integuments and has an opening called a micropyle. Inside the embryo sac are 3 antipodal cells, 2 synergids, a central cell, and the egg cell. An embryo sac is missing the synergids. What specific impact would you expect this to have on fertilization? A. The pollen tube will be unable to form. B. The pollen tube will form but will not be guided toward the egg. C. Fertilization will not occur because the synergid is the egg. D. Fertilization will occur but the embryo will not be able to grow. Sexual reproduction in gymnosperms. As with angiosperms, the life cycle of a gymnosperm is also characterized by alternation of generations. In conifers such as pines, the green leafy part of the plant is the sporophyte, and the cones contain the male and female gametophytes, figure 32.9. The female cones are larger than the male cones and are positioned towards the top of the tree. The small, male cones are located in the lower region of the tree. Because the pollen is shed and blown by the wind, this arrangement makes it difficult for a gymnosperm to self-pollinate. 896. Chapter 32. Plant reproduction. Access for free at openstax.org. Figure 32.9. This image shows the life cycle of a conifer. Pollen from male cones blows up into upper branches, where it fertilizes female cones. Examples are shown of female and male cones. Credit, female modification of work by geographer, Wikimedia Commons. Credit, male modification of work by Roger Griffith. Male gametophyte. A male cone has a central axis on which bracts, a type of modified leaf, are attached. The bracts are known as microsporophylls, figure 32.10, and are the sites where microspores will develop. The microspores develop inside the microsporangium. Within the microsporangium, cells known as microsporocytes divide by meiosis to produce 4 haploid microspores. Further mitosis of the microspore produces 2 nuclei, the generative nucleus and the tube nucleus. Upon maturity, the male gametophyte, pollen, is released from the male cones and is carried by the wind to land on the female cone. Link to learning. Watch this video to see a cedar releasing its pollen in the wind. Click to view content, https://www.openstax.org/l/pollen_release female gametophyte. The female cone also has a central axis on which bracts known as megasporophylls, figure 32.10, are present. In the female cone, megaspore mother cells are present in the megasporangium. The megaspore mother cell divides by meiosis to produce 4 haploid megaspores. One of the megaspores divides to form the multicellular female gametophyte, while the others divide to form the rest of the structure. The female gametophyte is contained within a structure called the archegonium. 32.1, Reproductive Development and Structure 897 Figure 32.10. This series of micrographs shows male and female gymnosperm gametophytes. A. This male cone, shown in cross-section, has approximately 20 microsporophylls, each of which produces hundreds of male gametophytes, pollen grains. B. Pollen grains are visible in this single microsporophyll. C. This micrograph shows an individual pollen grain. D. This cross-section of a female cone shows portions of about 15 megasporophylls. E. The ovule can be seen in this single megasporophyll. F. Within this single ovule are the megaspore mother cell, MMC, micropyle, and a pollen grain. Credit. Modification of work by Robert R. Wise. Scale bar data from Matt Russell. Reproductive process. Upon landing on the female cone, the tube cell of the pollen forms the pollen tube, through which the generative cell migrates towards the female gametophyte through the micropyle. It takes approximately 1 year for the pollen tube to grow and migrate towards the female gametophyte. The male gametophyte containing the generative cell splits into 2 sperm nuclei, one of which fuses with the egg, while the other degenerates. After fertilization of the egg, the diploid zygote is formed, which divides by mitosis to form the embryo. The scales of the cones are closed during development of the seed. The seed is covered by a seed coat, which is derived from the female sporophyte. Seed development takes another 1 to 2 years. Once the seed is ready to be dispersed, the bracts of the female cones open to allow the dispersal of seed. No fruit formation takes place because gymnosperm seeds have no covering. Angiosperms versus gymnosperms. Gymnosperm reproduction differs from that of angiosperms in several ways. Figure 32.11. In angiosperms, the female gametophyte exists in an enclosed structure, the ovule, which is within the ovary. In gymnosperms, the female gametophyte is present on exposed bracts of the female cone. Double fertilization is a key event in the life cycle of angiosperms, but is completely absent in gymnosperms. The male and female gametophyte structures are present on separate male and female cones in gymnosperms, whereas in angiosperms, they are a part of the flower. Lastly, wind plays an important role in pollination in 898 Chapter 32, Plant Reproduction. Access for free at openstax.org. Gymnosperms because pollen is blown by the wind to land on the female cones. Although many angiosperms are also wind-pollinated, animal pollination is more common. Figure 32.11. Angiosperms are flowering plants, and include grasses, herbs, shrubs and most deciduous trees, while b. gymnosperms are conifers. Both produce seeds but have different reproductive strategies. Credit A. Modification of work by Wendy Cutler. Credit B. Modification of work by Luz Castle UHI. Link to learning view and animation of the double fertilization process of angiosperms. Click to view content, https://www.openstax.org/l/angiosperms.